Good day, everyone. I'm Dr. Desiree Lucier, and welcome to my talk, Embedding Quality of Multi-Site Resting State fMRI Data in Clinical Cohorts Using Brain Parcellation. So just a quick background, MRI scanners use magnetic fields to generate images of the body. Functional MRI, or fMRI, takes advantage of the fact that increased neuronal activity elicits oxygenated blood flow to the active region. This leads to the blood oxygen level dependent or bold signal measured by fMRI both at rest and during task. During resting state fMRI acquisition, participants simply lay in the scanner in a state of rest. The typical eyes open paradigm for this is a crosshair in the center of the screen that participants are asked to look at for the duration of the scan. What we tend to see during resting state are some very specific networks of regions that appear to be functionally connected, giving us the functional connectome. Resting state functional connectivity is an emergent biomarker in Alzheimer's disease and other related neurodegenerative disorders. Specifically, consensus from group-level connectivity findings showed decreased connectivity in what is known as the default mode network in Alzheimer's dementia. In mild cognitive impairment, increased connectivity in the default mode and limbic networks were seen. Embeddings, which are representative of brain networks, are an important step in functional connectomics. However, it is currently unknown whether embeddings are able to represent different populations equally well, such as different ages or between men and women, clinical populations, or scanner manufacturers. One of the reasons that this is important is the increased need for and use of large-scale, large multi-site neuroimaging data sets, such as CIMAQ, for biomarker development, prediction of clinical trajectories, and neurodegenerative subtyping that could be useful for treatment purposes. A previous study investigated site and scanner differences on seed-based connectivity maps in the 7 common resting state networks obtained from a single Canadian subject, the single individual volunteer for multiple observations across networks, or the CIMAQ dataset. Side effects on map consistency were found to be highly heterogeneous. For within scanner manufacturer comparisons, for instance, when comparing 2 Siemens scans with each other, consistency was greater than between manufacturers, such as Siemens versus Philips comparison. Regions of interest in functional connectomics are obtained using brain parcellations. There are many different atlases and types of functional brain parcellation. In general, these atlases consist of either hard parcellations or soft parcellations. Hard parcellations have regions which are binary, where a voxel is either part of a region or it is not. Soft parcellations have regions with probabilistic maps. So instead of a 1 or a 0, there is a sliding scale membership. Current functional imaging parcellation techniques have limited homogeneity, with average correlations between voxel level time series and parcels ranging between 0.5 and 0.6 for many popular atlases. However, work from our lab found that using dynamic instead of static parcellations can generate highly reproducible seed-based brain parcels at the individual level. Further extending this method to the full brain, dynamic parcel aggregation with clustering, called DIPAC, is a Python package capable of detecting stable whole brain dynamic parcellations and fMRI data by using a simple 2-level clustering algorithm. For the first level, k-means is run directly on the time series using sliding time windows. This generates a family of parcels which are represented with 1-hot encoders. The second level performs clustering on indicator function of parcels aggregated over many windows. This procedure gives the stable maps. Neuroimaging data was pulled from multiple large neurodegeneration and Alzheimer's databases, CIMAQ, CCNA, and ADNI, as well as the CIMAQ dataset, which consists of a single healthy Canadian subject scanned at multiple sites over time. Datasets were pre-processed using a standard fMRI pipeline implemented in the Neuroimaging Analysis Kit version 1.1.4, or YAC. The pre-processing procedures were applied: slice timing correction, estimation of rigid body motion in fMRI runs, both within and between sessions, co-registration of the structural scans in stereotaxic space, individual code registration between structural and functional scans, and resampling of functional scans in stereotaxic space. DIPAC was then run using the resampled functional resting state data and confounds. Model parameters consisted of 64 clusters, 1,024 states with 8 mm smoothing, and were run with the MNI gray matter template. 80% of the CIMAQ, CCNA, and ADNI data were used for training, with the remaining 20% and CIMAQ dataset combined for testing. Individual subject time series from the test set were used to generate R-square maps from the models to quantify embedding quality. For these R-square maps, a score of 1 is a perfect approximation and 0 is none, with a negative score being a very poor embedding. NISTATS was used to compare R-square maps to evaluate for significant differences in embedding quality between datasets, sexes, ages, clinical diagnoses, and scanner manufacturer with an alpha of 0.05. FDR correction was then applied to each comparison. Nylern plotting was used for visualization of the R-square maps and contrast results. Consistent with the earlier mentioned study, scanner manufacturer comparisons revealed significant differences between Philips and Siemens, Philips and GE, and Siemens and GE. There were no differences in average embedding quality between the CIMAQ, CCNA, and CIMAQ datasets. However, there were differences in average embedding quality between ADNI and the other datasets. It should be noted that CIMAQ, CCNA, and CIMAQ are using a harmonized scan protocol. This likely contributed to a reduction in embedding quality variance between those datasets. Additionally, variation in scanner manufacturer ratios within datasets may have contributed to the differences seen in embedding quality between ADNI and the others. The test sets for CIMAQ and CCNA, which show the greatest differences with ADNI, had a larger proportional number of scans acquired on Siemens scanners. The ADNI set, on the other hand, had a proportionally larger number of Philips scanner acquisitions. There were no differences of embedding quality with age in a regression model. There were also no differences in embedding quality between women and men, or between most clinical diagnoses, including between adults considered cognitively healthy and those with subjective cognitive impairment or mild late cognitive impairment, between subjective cognitive impairment with mild late cognitive impairment or Alzheimer's dementia, or between mild late cognitive impairment with Alzheimer's dementia. However, there were significant differences in the quality of mean embeddings between adults considered cognitively healthy and those with Alzheimer's dementia or early mild cognitive impairment, between subjective cognitive impairment and early mild cognitive impairment, and between Alzheimer's dementia and cognitive impairment, early cognitive impairment. Again, this may be due to differences in embedding quality between datasets resulting from other previously mentioned factors. Future plans for DIPAC include expanding these findings to other heterogeneous multi-site fMRI datasets, including developmental and psychiatric populations, and the investigation of scanner upgrade and site-specific effects. We are also planning comparisons of DIPAC with more traditional independent component analyses and other parcellation methods. Additionally, we plan to investigate the effects of different parameters and resolutions on embedding quality to optimize our models. In conclusion, this has been an important early step for the validation of DIPAC for dimension reduction and heterogeneous multi-site data. Embedding quality was consistent for sex and age. DIPAC also scaled well for big data. Scanner differences were consistent with the literature suggesting systematic bias in embedding may result in site differences. Additionally, the results of the dataset comparisons emphasizes the importance of protocol harmonization between scan sites. Overall, DIPAC has the potential to increase the robustness and scalability of resting state fMRI as a biomarker in neurodegeneration, especially in the context of big data multi-site acquisition. I would like to say a quick thank you to my mentor, Professor Pierre Belec, Professor Aman Benoit, Dr. Natasha Clark, Hanan Schmark, the CIMAQ Lab, Simone, CIMAQ, and everyone for being here today. Thank you.